For our project, the first thing we had to do was figure out what species were capable of generating current. So we did a literature search and we found that these two species, Geobacter sulfurreducens and Shewanella onidensis, were both capable of doing so. Geobacter has metallic-like pili in terms of its conductive mechanism. That just means that the pili conduct electrons internally, basically just through the structure. They reduce organic compounds like carbon dioxide and acetate to produce a current. Uh, what's also kind of cool about them is that they're capable of forming aggregates with other bacterial species. So just basically think about a biofilm that can metabolize these compounds and then produce a current within itself. They accept these electrons from methanogenetic metabolism in these biofilms via the pili, and there are known strains, KN400 to be specific, that produce more current. Genomic comparison with this strain didn't really detect any novel genetic basis for this. It seems like this is linked to just increased ATP production. It is an obligate anaerobe, which means it would be pretty hard to work with functionally. Shewanella reduces metals to produce a current, and it uses an electron hopping mechanism on its pili, which is slightly different than the Geobacter. One of the first concepts we considered for our project was a fuel cell. Basically, the way that one of these fuel cells would work is you have one of these species metabolizing its substrate. It would produce a current. You attach that to an electrode, and in the diagram over here, you can see that, I guess, powering a calculator. And what we were thinking about doing was trying to figure out a way to make these fuel cells more efficient. So there are a number of ways we can do that, at least what have been hypothesized in literature, was to do something beyond just increasing the conductivity of the pili, perhaps making a matrix that would kind of conduct electrons between pili rather than a long pili. The scope we figured out was a little bit too big for 6 weeks. There's a lot of variables that we would have to consider, electronic engineering variables, chemical variables, scalability. Because of the scope of the project so big, and there's a lot of existing literature on the subject already, we decided to move on to other options. One of the next concepts that we considered was using a different chassis. There's a little bit of newer research going on with a strain called Vibrio nitrogens. This is a halophilic strain. It means it loves salty environments. And it turns out it divides extremely fast. So on the figure here, you can see a growth rate comparison between Vibrio and different strains of E. coli, some of the more faster growing strains, and you can see it grows much more quickly. It's also extremely modal, which might be useful in different applications. The genes that we are already aware of, and Shewanella and Geobacter engineered into Vibrio, could make for some new interesting technology. What would also be kind of cool about this is we would be able to characterize Vibrio a bit more in terms of its clonability and its ability to uptake new genes. Some of the Vibrio drawbacks that we ran into were right off the bat the halophilic nature of the species. Initially, it was thought that maybe we could use this constraint to think up some interesting applications, maybe electronic repair in saltwater environments, things like that. But it turned out that this is going to be more of a hurdle than a help. So specifically, Vibrio nitrogens is not as harmful as some other species of Vibrio. The figure here, which I chose a little bit more of a gentle figure, there's some pretty nasty ones out there. This shows an infection of Vibrio. Yeah, it can infect a person, and it can kind of turn into a flesh-eating bacteria, so not so fun. The other thing is that it isn't very well characterized, so if we had more time, it would be good to maybe do some work and help characterize it a little bit more, but as it stands, this is going to be, again, more of a hurdle for us. The next idea we moved on to was material synthesis, trying to use some form of bacteria to generate these nanowires for materials production. Potentially, there could be a lot of really good applications out of this. One of the ones that jumped immediately to mind was solar cell materials. If we could create a vertically aligned forest of conductive pili, or sometimes they're called nanowires, we could potentially come up with some new ideas that would improve over the existing materials that are out there, basically tin oxide. So if you notice on the diagram, there's an anti-reflection coating right underneath the glass cover. This is to make sure that the photons that come in don't get reflected back off the material that's supposed to be conducting them down and generating current. We had two potential ways to do this. One would be cell-based. The cell-based has some good benefits in that the pili could potentially have a support system. If the pili got damaged, the cell could potentially produce more, maybe even be programmed to have a certain density. Part of the drawbacks of this would be trying to engineer the cell to generate these pili vertically, unidirectionally, and maybe even polarly, so you have basically cells producing pili that come out the top and the bottom between two electrodes. There's another phage-based method. This had some benefits in that there are already some existing literature and methods out there, and because they're a lot smaller scale, they would have a higher level density of these nanowires. Some of the existing methods that we found were vertically aligned phage tails. Basically, these researchers would produce these tails. They would process them in a way that would align them vertically, and they would end up being piezoelectric, which means that mechanical stress would induce a current. This was pretty cool. They would be able to do things like implant these in pacemakers and have the beating of the heart actually recharge the pacemaker. In terms of materials application, for the solar cell, you'll notice that the diameter is about 6 nanometers. The shortest wavelength of visible light is something more like 280, 290 nanometers. So it's well below that threshold. So we wouldn't have to deal with that reflectivity problem that you noticed in the last slide with the anti-reflective coating. The problem with this is that the processing to get this to happen is well beyond the scope of what we're capable of in 6 weeks. There's a nanopore filter that needs to be either fabricated or ordered just to get these things aligned vertically, and the technical experience necessary to do this is, I think, beyond what, again, we could manage in such a short time frame.